Hello students. In this video, we are going to see about antibiotics, their classification based on their mode of action, based on their structure. We are going to discuss in this particular video. So these are the contents that we are going to see in this video. So as we know, antibiotics are nothing but the substance that inhibit the microorganisms or the substance which kills the microorganisms. So suppose if the substance inhibit the microorganism, then it is called as bacteriostasis. And if the substance kills the microorganism, it is called as bactericidal. This is Alexander Fleming. In the year 1928, he first discovered the penicillin, which laid the foundation of antibiotic chemotherapy. And some other examples for antibiotics include cephalosporin, chloramphenicol, and so on. So what happens if there is an abnormal use of or overuse of antibiotics? Both in human as well as in medicine, it leads to the formation of superbugs. Superbugs are nothing but bacteria which doesn't have any influence on the conventional antibiotics. Even though if we consume much antibiotics for a particular kind of disease, that particular bacteria has no effect on that particular antibiotics. That is called the superbugs. And these antibiotics are classified based on three categories. One is based on structure, based on function, and another one is based on spectrum of activity. So first we see about the based on function category. So in the first case, it is the inhibition of bacterial cell wall synthesis. So normally the bacterial cell wall, it is made up of peptidoglycan, which is a polymer, and it has a cross-linked polypeptide chain. So this consists of N-amino glucosamine, N-acetyl glucosamine and N-acetyl murmuric acid. So these two combines together to form the peptidoglycan, so which is mainly responsible for the flexibility of the cell membrane. As we know, all the microorganisms are considered as prokaryotic organism. Why it is considered as prokaryotic organism? Because it doesn't have a nucleus. They are considered as unicellular organism and it doesn't have a nucleus. So thus it is made up of a peptidoglycan uh, glycan. And this is activated by an enzyme which is nothing but transpeptidase enzyme, which is also called as penicillin binding protein. On the other hand, we have beta lactam, which is mainly responsible for the antibacterial activity. So when this beta lactam is open, then the antibacterial activity is lost. So this beta lactam will mimic the structure of D-alanine terminus of peptidoglycan precursors. So owing to this, there is a the enzyme becomes inactivate. That is, the transpeptidase enzyme becomes inactivate. This leads to the formation of osmotic lysis and bacterial cell death, which means osmotic lysis are nothing but the rupture of the cell membrane. When the cell membrane is ruptured, automatically cell death will take place. And we know that this bacteria can be classified into two types. One is gram positive, another one is gram negative bacteria. So here in case of gram negative bacteria, we have an outer membrane. So through this outer membrane, the beta lactam will penetrate and binds directly with the penicillin binding protein, which helps in the cross-linking of peptidoglycan, which is mainly responsible for the bacterial cell wall formation. On the other hand, gram-positive bacteria, it doesn't have an outer membrane. So, beta-lactam will directly bind with the uh, penicillin protein binding, and thereby the peptidoglycan synthesis process is being completely affected. So if this is affected, then the cell death process will take place. Then the inhibition of protein synthesis. Bacterial proteins such as 70S ribosomes Uh, it consists of 30 S small subunits and 50 S larger subunits. So here, this is the picture for the 70 S ribosome. Here at the bottom, you have the 30 S subunit, and at the upper, you have the 50 S subunit. So what is the role of this 70 S ribosome is to transfer the mRNA to the rRNA. So, this process will enable selective toxicity. Now, these are some of the examples like tetracyclines, aminoglycosides, macrolides, linezolid. All these are some of the examples for the protein synthesis. So, when we talk about the tetracyclines and aminoglycosides, they binds with the 30 years ribosomal unit in the reversible and irreversible manner. Reversible manner in the sense these 70S ribosomes can easily dissociate to form 30S and 50S ribosomal units. Whereas in case of irreversible manner, the 70S ribosomal cannot be dissociated only either by certain reaction mechanisms or certain mode of action, the dissociation will take place. Okay, then comes the macrolides and linezolide. These will bind with the 50S ribosomal unit, and both are considered as the bacteriostasis agent. Why it is considered as bacteriostasis agent? Because it doesn't cause the cell death, whereas it inhibits the growth of the microorganisms. So other than these proteins, we have another protein which is nothing but chlorpheniramine, which is also an antibiotic. The next comes the metabolic pathways, disruption of cytoplasmic membrane, and inhibition of nucleic acid synthesis. So, in case of metabolic pathways, inhibiting the synthesis of essential metabolites like folic acid, mainly this folic acid is important for women in case of ovulation process, and humans will consume the folic acid through the diet. Okay, so this folic acid metabolites have been inhibited with the help of bacteria. So, in order to avoid that, we used to take antibiotics. The next one, disruption of the cytoplasmic membrane. So the disruption will take place by selective permeability process, where it allows only certain ions to pass through the membrane by effluxing the other ions and influx the extracellular fluids like water into the membrane. So this will causes the rupture of the cell membrane, thereby cell death arises. And the last category is the inhibition of nucleic acid synthesis. Several bacterial enzymes like DNA gyrase and topoisomerase 4, all these are employed for supercoiling and replication of DNA purpose. So this can usually cause a bactericidal process leading to the destruction or death of the cell. And the next one is based on the spectrum of activity, it is classified into two categories. One is broad spectrum, another one is narrow spectrum. So, from the name itself, it is clear that if it is narrow spectrum, it fights against either gram positive or gram negative. That is, it fights against only a selective or a specific microorganisms, which means if the person is infected by some kind of pneumonia virus, this particular antibiotic will directly uh, works on that particular particular cell, infected cell, rather than the other host cells, unaffecting the host cells. That is called as narrow spectrum. And in case of broad spectrum, this will fight against both gram positive as well as gram negative bacteria. That means it will fight against numerous types of bacteria. And apart from that, the mechanism of action also plays a vital role. So these bacterial agents inhibits the synthesis of bacterial cell wall, and it also directly binds on the cell membrane of the microorganisms, acting as a selective permeability. And this will affect the functions of 30S as well as 50S ribosomal subunits to cause a reversible inhibition of the protein synthesis. Then comes the classification of antibiotics based on chemical structure. So it is mainly based on beta lactam group. Beta lactam group is nothing but the which is mainly responsible for the antibacterial activity. So this group is further classified into four main categories. One is penicillin, another one is cephalosporin, another one is mm, carbapenem, and another one is uh, monobactam. So, in case of penicillin, penicillin exhibits excellent activity against both gram-positive as well as gram-negative bacteria. And in case of cephalosporin, it has a dihydrothiazidine ring and it has a different classification based on the generations. Mainly five generations are involved. All those five generations are involved to fight against both gram-positive as well as gram-negative bacteria, and it also fights against the methicillin-resistant Staphylococcus aureus bacteria. And in case of carbapenem, this particular bacteria is coming under broad spectrum, whereas penicillin and cephalosporin comes under narrow spectrum. This will comes under broad spectrum because it fight against different types of microorganisms. That's why it is coming under a broad spectrum category. And in case of monobactam, this contains a beta lactam ring which fight against the gram-negative bacteria. The next one is based on the enzyme-based mechanism of action. Several enzymes also plays a vital role against the bacterial infections, such as beta-lactamase enzyme, which have the ester as well as the amide bond. Say, for example, penicillin, you have the ester as well as the amide functional group. This can be further classified into four categories. Class A beta lactamase, class B beta lactamase, class C beta lactamase, and class D beta lactamase. So, in case of class A beta lactamase, they are mainly found in Enterobacteriaceae, which are designated as STEM1 and SHB1. So this is actually named, this name comes from the patient from whom it was first isolated. So, and SHV is also a self-hydral variable, which is mainly involved uh, uh, for the which is mainly considered as a non-extended spectrum of beta lactase. So this will act as an precursor for the extended spectrum of beta lactase. And next one is class B beta lactamase. They are metallo-beta lactamase, and metals such as zinc or heavy metals used as a catalyst will activate this particular enzyme, and these are inhibited by the chelating agents. And in case of class C lactamase, they are also called as cephalosporinases. They are produced by gram-negative bacteria, and they have large cavities when compared to the class A and class B category. And in case of class D beta lactamase, one fine example is oxacillin, which is also found mostly in Enterobacteriaceae. The next category is aminoglycoside modifying enzymes, and the last one is chloramphenicol acetyl transferase. In case of aminoglycoside modifying enzymes, they are mainly categorized as affinity based enzyme because they reduce the modified molecule by binding to the 30S ribosomal subunit. And in case of chloramphenicol acetyl transferase, certain gram-positive as well as gram-negative bacteria and some of the other virus or bacteria are resistant to chloramphenicol, and they have an enzyme uh, that acetylates hydroxyl groups of chloramphenicol, and this will leads to the disruption of the 50S ribosomal subunit, and this will directly inhibits the formation of the bacterial sites, which is also considered as the staphylococcus bacteria.